Hello everyone, this is Medical Gateway, and today we're going to start a new chapter, which is chapter number 3 of Guyton, the genetic control of protein synthesis, cell function, and the cell reproduction. So let's start. Okay, in this part, we are basically going to study that how exactly, what is the structure of the DNA and how exactly RNA is formed from DNA. And in the next parts, we will study that how exactly the RNA results into the production of the proteins. So let's start it. Okay, so in the first slide, the basic overall concept of that how exactly the DNA controls the cell functions. DNA controls the cell functions by protein formation, and that protein will act as structural proteins as well as functional proteins, which are basically enzymes. So structural protein will form the structure of the cell, and functional proteins, the enzymes, will control the functions of the cell. So that's how DNA controls the cell function by the formation of protein. Now, how exactly DNA results into the formation of the protein? DNA results in the formation of RNA, and then RNA will cause the formation of protein. The formation of RNA from the DNA is known as transcription, whereas the formation of proteins from the RNA is known as translation. Okay, so what is transcription and what is translation? The formation of RNA from DNA is transcription, whereas the formation of protein from RNA is translation. Okay, so what exactly is happening? This is the whole cell, and this is the nucleus. So inside the nucleus is the DNA. DNA is double-stranded. Now DNA will be converted into single-stranded RNA. Then splicing of this RNA will take place, and this RNA will be converted into its subtype, the messenger RNA. Now this is the messenger RNA. Now this messenger RNA will go out of the nucleus into the cytoplasm, and there this messenger RNA will go to the ribosomes, these orange colored ribosomes, and on ribosomes this RNA will result into the production of protein, which is translation. So basically, there is a genetic code in DNA. That genetic code will be transferred to RNA, and that RNA will be converted into messenger RNA. It's one of its subtypes. That messenger RNA will carry a code. Code for what? Code for the protein formation. Code for the sequence of the amino acids in the protein formation. So this code will define that which amino acids are going to be attached in what sequence. Okay, and then protein will be formed. So this is how DNA results into RNA, and RNA is converted into proteins. Now in the whole chapter, we'll discuss in detail the process of transcription and translation. Okay, now what is gene expression? Now gene expression is basically the conversion of DNA into RNA and RNA into protein, or the transcription and translation is collectively known as gene expression. And what is gene? The gene is, in simple words, it is basically a specific sequence of nucleotides collectively form a gene. Okay. Now, how many genes are present in a DNA? So there are 30,000 different types of genes, which will result into 100,000 different types of proteins. So around 3 to 3.3 times proteins are produced as compared to the genes. Okay, so this is it. So now let's discuss in detail the double helical structure of DNA and how exactly DNA is going to produce RNA. So let's start. So DNA is a double helical structure as shown here. So this is one strand of DNA, and this is the other strand of DNA. So this one strand and the other strand, there is a bond between these two strands, and then these two strands are present in the form of this double helical structure. Okay, now what is the basic unit of a DNA? The basic unit of a DNA is a nucleotide. Now, what is a nucleotide? Nucleotide is basic nucleotide consists of three things. Number one, this phosphoric acid with the P, and then D is this deoxyribose sugar, and then the third one is the nitrogenous bases. So for DNA, the nucleotide consists of, in the DNA, phosphoric acid, deoxyribose sugar, and a nitrogenous base. Phosphoric acid, deoxyribose sugar, and a nitrogenous base. Now the nitrogenous, okay, now so this is a nucleotide of a DNA. So if there is a nucleotide of RNA, then what is the difference? RNA also has the same phosphoric acid, it also has the nitrogenous base, but instead of deoxyribose sugar, RNA has a ribose sugar. Okay, so in place of this D, in the case of RNA, this D will be replaced by R, ribose sugar. So DNA has deoxyribose sugar and RNA has ribose sugar. So this is one difference between DNA and RNA. The other difference is DNA is a double helical structure. It has two strands, and RNA is single stranded. It has only one strand. Okay, and the third difference is in the nitrogenous bases. Now let's discuss nitrogenous bases, and then after that I'll tell what exactly is the difference in the nitrogenous bases of DNA and RNA. Now this nitrogenous bases, there are two broad types of nitrogenous bases. One is purine and the other one is pyrimidine. So purine is this A and G is purine, and pyrimidine is T and C. So this is A is adenine, G is guanine, T is thymidine, and C is cytosine. So purines are adenine and guanine, pyrimidines are thymine and cytosine. Okay, now interestingly, purines of one side will form bond with the pyrimidines of the other side. So this adenine and guanine are purines, they're forming bond with thymine and cytosine. Okay, so purine will form bond with the pyrimidines of the other strand. Okay, so exactly, adenine will form bond with thymine and guanine will form bond with cytosine. As you can see here, adenine is forming bond with thymine and guanine is forming bond with cytosine. Here thymine is forming bond with adenine and cytosine is forming bond with guanine. So this is how the purines and pyrimidines are arranged and how they form bond. Okay, so the bond between them is the hydrogen bond. This is not a covalent bond, this is a hydrogen bond. The bond between the two nucleotides, this is one nucleotide and this is other nucleotide, the phosphoric acid, the deoxyribose sugar, and the nitrogen base, this is the other nucleotide. So the bond between the two nucleotides, this bond, this is a covalent bond, whereas the bond between the two chains or the nitrogenous bases is hydrogen bond. Okay, now I just mentioned that there is difference between the nitrogenous bases of RNA and DNA. Now what exactly is the difference? In the case of RNA, all the three bases, adenine, guanine, and cytosine, are present, but instead of thymine, RNA has uracil. Instead of thymine, RNA has uracil by the letter U. Okay, so this is the double helical structure of the DNA. Okay, now when the DNA will form RNA, what is going to happen? This double helical structure of the DNA will be split into two single strands like this. When the DNA is going to form RNA, the first step is that this hydrogen bond between the two strands is broken, and these two strands will be separated from each other just like this. So this is now a single strand of DNA, and these are its nucleotides: this phosphoric acid, deoxyribose sugar, and the nitrogenous base, and then another nucleotide, and then another nucleotide. Now the bases are these: thymine, cytosine, adenine, cytosine, guanine, and guanine. Okay, now this strand will produce single-stranded RNA chain here. And how exactly they are going to produce? It's pretty simple. the RNA bases will come and attach to these DNA bases, or the RNA nucleotides having the counterpart bases will come and attach to these nitrogenous bases of DNA nucleotide. For instance, if this is a nucleotide and its nitrogenous base is thymine, now which RNA nucleotide will come and get attached here? Thymine is always attached with adenine. So an RNA nucleotide having adenine will come and will be attached with this thymine, but it has few other steps before the attachment. So how it's going to take place? So this RNA nucleotide having this phosphoric acid, this ribose sugar, R instead of the deoxyribose sugar. Now there is adenine as the nitrogenous base. Now this nitrogenous base will be attached with this thymine of the DNA nucleotide. So this is an RNA nucleotide. Okay, now first step is this RNA nucleotide will be activated. There will be activation of this RNA nucleotide. And how this RNA nucleotide is going to be activated? It will be activated by the addition of two high energy phosphate bonds. Sorry. So these two high energy phosphate bonds will be attached with the phosphoric acid of the RNA nucleotide. And who will attach these bonds, high energy phosphate bonds, with the RNA nucleotide? That is the enzyme RNA polymerase. Now this rounded green structure is the RNA polymerase. Now RNA polymerase will basically activate this RNA nucleotide by the addition of two high energy phosphate bonds. And when this nucleotide is activated, there will be formation of hydrogen bond between the DNA nucleotide and the RNA nucleotide, or to be specific, the counterpart nitrogenous bases of the DNA nucleotide and the RNA nucleotide. Okay, so this is how it is attached. Okay, now, and this will happen to all the nitrogenous bases of the DNA, and their counterpart nucleotide, RNA nucleotide, will be attached in the form of a chain. Okay, for instance, there it is, cytosine. This will be attached by RNA nucleotide having guanine as the nitrogenous base. Okay, so this step is, I've shown you just this step and this step. Actually, the whole chain of the RNA is already formed just like this. So here it was guanine, then cytosine, then guanine again, cytosine. For cytosine, there is guanine. For adenine, there is uracil, not thymine. For guanine, again, there is cytosine. For adenine, there is uracil. So this is the complete chain of RNA which is being formed, Okay, now, when it is activated, the bond will be formed between them. After that, what is going to happen? A bond will be formed between the two adjacent RNA nucleotides. Now, a bond will be formed between the phosphoric acid and the ribose sugar of these adjacent nucleotides, just like this, as the bond is formed here between this phosphoric acid and this ribose sugar. And this bond will be covalent bond. This is the hydrogen bond, and this bond will be between the two adjacent nucleotides as the covalent bond. And when this covalent bond is formed, then this nucleotide will get rid of these high energy phosphate bonds. The main reason of these high energy phosphate bond was the activation of the RNA nucleotide. Now the whole job is done. These high energy phosphate bonds will be separated, and the whole RNA chain will be formed in this form. Okay, and after that, when the covalent bond is formed and the two high energy phosphate bonds are detached, it will then result into breakage of this hydrogen bond. We don't need this hydrogen bond. And finally, the whole RNA, single strand of RNA chain will be formed from the DNA, and exactly the DNA will produce the counterpart bases, the counterpart nucleotides chain exactly in the same sequence as the DNA strand has. Okay, so this is how the RNA strand is produced. Now, a few other points in this story. Now, if we focus here, what exactly is a code triplet in a DNA? So, a code triplet is basically a sequence of 3 nucleotides, or to be specific, a sequence of 3 nitrogenous bases. Now, these 3 nitrogenous bases, this will form a code triplet, and the code triplet is formed in the DNA, or simply a code. Okay, now this code will basically define that which protein will produce. This code will define the code of the RNA, and this code of RNA will define which protein is going to be, which amino acid in the sequence is going to be attached. Okay, and which protein is going to be produced by the addition of the amino acids. Okay, so this is a code triplet of the DNA. Now, if you focus on this and on this RNA chain has started to form from this point, and then it will keep forming, and at this point, this RNA chain will end. So the point where the RNA chain is going to be formed from the DNA, we need something to promote the formation of RNA from the DNA. So this initial sequence of bases is known as the promoter sequence. So this initial sequence of the bases is known as the promoter sequence. This sequence will promote the start of the RNA production from the DNA strand. Okay, now if we have a promoter sequence, then at the end where we need termination of this production of RNA chain, there should be some chain terminating sequence. So at the end, for instance, if this nucleotide, this nucleotide, and this nucleotide, these 3 bases will combine to form a chain terminating sequence. It means that wherever this sequence is present, the RNA will start to be produced from this sequence, and wherever this sequence, the chain terminating sequence, is present, there the production of the RNA will be terminated. The production of the chain of the RNA will be terminated. So this is the promoter sequence, and this is the chain terminating sequence. At the start of the DNA, from where the replication is starting, the production of DNA is starting, that is promoter sequence, and where it is ending is the chain terminating sequence. Okay, now this, now coming back to the code triplet. The code triplet is present on the DNA, whereas its counterpart nitrogenous bases GGC will result into the production of CCG on the RNA. So these nucleotides and these specific sequence of nitrogenous bases is known as codon. So this was code triplet on DNA, and this is the codon on RNA, to be specific on the messenger RNA, because messenger RNA is produced in the nucleus, and that has to take the message to the cytoplasm on the ribosome for the production of proteins. Okay, now, so this is the code triplet on DNA, and this is the codon on the messenger RNA. Okay, now this is one RNA nucleotide, this is the second RNA nucleotide, this is the third RNA nucleotide. These 3 will combine to form a codon. Okay, now what is the function of this codon? This codon is going to define the amino acids. For instance, now this messenger RNA comes out of the cytoplasm, comes out of the nucleus and goes to the cytoplasm, and then in the cytoplasm, this codon will define that which amino acid is going to get attached in the sequence. So the story starts here. If there is the sequence CCG, it means that amino acid proline is going to be attached first. Then we have the sequence UCU, which is here UCU. UCU means that after proline, serine amino acid will be attached with the proline, and then the sequence GAA. This means that after serine, glutamic acid will be attached. Now this amino acid proline, then amino acid serine, and then amino acid glutamic acid. These all will bind to each other in the chain, and this chain will form a protein, a specific protein with the specific sequence of amino acids. So this is how exactly the DNA, the genetic makeup in the DNA, the specific sequence of nucleotides in the DNA, and to be precise, the specific sequence of the bases or the code on the DNA has produced a specific amino acid by initial production of messenger RNA, and which has carried the message or to the ribosomes for the specific attachment of different amino acids in a sequence. Now, this specific protein might act as a functional protein in the form of enzymes, and this can increase or decrease or move reactions in different directions. This is pretty important. Or this can result into the formation of some structural protein. So this is how the DNA is basically controlling the cell function by the production of the proteins. Okay, so this is it. In this part, we have basically discussed DNA's structure and how exactly RNA is formed from the DNA, which is transcription. In the next parts, we will discuss that exactly how this code is leading to the formation of these amino acids in serine, and how the transfer RNA are carrying these amino acids, and how transfer RNAs are reading this code of messenger RNA, and what is the role of ribosomes, and what are the types of ribosomes, and things like that. So basically, we have discussed transcription here, and in the next parts, we will discuss translation. See you in next parts.